We are recording. It's about 12 minutes before the lecture actually starts. The first word for attendance is going to be court. C-O-U-R-T, court. So if you're watching the video, that is your first word for attendance. As always, you're going to need to take screenshots every time I say one of the attendance words or phrases. As always, you're going to need to email me that list of words and a screenshot for each time, again, that I said the word. And as always, the last attendance phrase will be you asking me a question about the material presented in today's lecture. And that is it. So, like I said, it's about, well, at this point, 11 minutes before we start. So if you want to, feel free to skip ahead in the video about 10 to 11 minutes. You have a good weekend?
Yes, sir.
Good.
Where's the weekend?
Average, I guess, which is a good thing. Nothing,
Yeah.
nothing bad happened.
You been watching March Madness?
No, I mean a little bit. My in-laws went to University of Tennessee, so I was watching that for that reason. I hate Florida, so I like to watch those games to see,
Yeah.
But other than that, not really. How about you?
Yeah. I've been really tapped into it.
I noticed, I don't remember which game it was, but they were playing in Indianapolis. I was like, what the hell? The court looked to be elevated. So I looked it up, and they were playing in a football stadium. I had no idea. So they had just brought the court in and put it in the middle of the stadium. That was neat.
Yeah, I'm about to say, most of the games is like at neutral sites.
Right.
So they put them in like football stadiums and like one team, I think the Purdue game, they was playing in say they were playing in Ohio.
Oh, yeah, David.
Yeah.
So,
Morning. I try to get into it. I don't know. It's not into basketball
Yeah.
or professional football. And I love college football. I just can't get into professional football. I try every year.
Yeah.
Just having an extra day or two of football would be amazing, but I don't know. Probably have to go to a game. That would change things. Because I liked college football, and so I went to a game, and then I loved it.
Yeah.
Once I went, I was hooked. So maybe that's what I need to do.
Can you open my quiz for me again? I can put this card.
Yes,
I got a bad grade the first time.
if you don't mind, send me an email. Remind me. All right, we've got less than a minute left. I'll do attendance. Again, if you're online and I call your name and say you're not here, don't worry about it. Just send the words in before 9 a.m. Bangura is not here. Case Bolt is here. Trisden is not here. Covington Driggs is not here. De Beer, wait, never mind. Dow is here. Hughes is not here. Lebdy is not here. Carl is not here. Mankins is here. Mathis is here. Rains is not here. Reed is not here. Elise is not here. Thompson is not here. Withrow is not here. Yindui is not here. And Elkins is not here. So 1, 2, 3, 4. I got the 4 people who are physically here. Again, the rest of you, business as usual, send in the attendance words before 9 a.m. And of course, if you're watching the video as opposed to the live stream, you're going to send a screenshot of each time I say one of the attendance words. So not just the attendance words, but also screenshots. And of course, for those of you who are physically here, you don't have to send those words, but you can if you want more extra credit. All right, let me back up. I'm not going to do announcements yet. Let's go back to where we left off, which is this overview. And again, my recommendation is to memorize the big picture and then start adding the details. Depending on how your brain works, everybody learns differently, but in my opinion, that's usually the best way to learn this. So first of all, like I mentioned last week, the first thing to memorize is the fact that we go from DNA to RNA to protein. Right, memorize that first. Then start memorizing some of these facts that I'm about to teach you. And again, what we're about to do is an overview. It's sort of like how we did it for photosynthesis and respiration, where we had an overview, and then we went back and kind of looked at the details. I'm doing it the same way. And speaking of which, to remind you, this is different than your textbook, right? Your textbook doesn't do it this way. This PowerPoint is still based on the old textbook we used to use. And in this particular instance, I think the old textbook was superior to the new textbook. So this is why I'm sticking with that. Anyway, again, the information that is written in the nucleotides of the DNA is transcribed into a molecule of RNA. And then that molecule of RNA is then translated into polypeptides, which are then folded into proteins. So again, big picture, memorize that first. Memorize the fact that we go from DNA to RNA to proteins. Then once you have that memorized, then memorize what it's called, right? Going from DNA to RNA is called transcription. Going from RNA to protein is called translation. And later, when we get into more details, I'll give you a way to help remember the difference between transcribe and translate. But again, this is still just the overview. The first word for attendance will be the last word in this bullet point. So what is the last word in that bullet point? First word for attendance. Any questions about this slide? The next slide I'm going to show you is a very simplified picture of this process. Again, we're just looking at the overview. We're not going into details yet. So here we have a strand of DNA inside the nucleus. You can see outside of the nucleus is the cytoplasm. So that's just the cell that's in blue. The nucleus is in purple. That DNA, the code in the DNA is transcribed, right? We take that code that's written in the DNA and we use it to make a molecule of RNA. That process is called transcription. The RNA goes through some processes that we're going to talk about later, at which point it leaves the nucleus, and then it is translated into protein. So again, the code starts in DNA. We use that code to make a molecule of RNA. That all happens inside the nucleus, which is a great test question. Where does transcription happen? Inside the nucleus. Anyway, the code in the DNA is transcribed, right? We use that code to make a molecule of RNA. The RNA goes through some processes that we'll talk about later. It then leaves the nucleus and goes into the cell, into the cytoplasm, where that code is then used to bring a bunch of amino acids together, which builds a protein. And that process is called translation. And again, that's another good test question. Where does translation happen? In the nucleus or in the cytoplasm? And translation happens in the cytoplasm. So that will definitely be, those are two definite test questions there. Where does transcription happen? Where does translation happen? Also, what does transcription make? RNA. What does translation make? Protein. Okay, a lot of test questions in that one simple picture. Any questions about that picture? All right, if you download the PowerPoint, you can watch this animation that shows translation happening. It shows protein synthesis. I recommend watching it. Again, these videos they're made for higher level students. So if you see any words in those videos that he didn't teach me that, don't worry about it, right? You don't need to know those words. Anyway, so I keep using this word transcription and translation, right? So in a sense, we're talking about a language. So then that brings up the question, what is the language of nucleic acids? And the language was the nucleotide bases. Like I mentioned earlier, when we were talking about DNA and the structure of DNA, I was saying it's those nucleotides, that's the code. The A's, the C's, the T's, and the G's, or if you're talking about RNA, there's no T, whoops, excuse me. If you're talking about RNA, there's no T, there's a U instead. But either way, those are the four letters in this language, right? So when we transcribe, hopefully this will help you remember, when we're transcribing, it's the same language, right? We're going from DNA to RNA. So we're going from one type of nucleic acid to another type of nucleic acid. That's how you can remember, I hope this will help you remember, that it's the same language, right? So we're not translating anything. We're not going from one language to another. It's the same language. So you're just writing down the message. And if you know what that means, that's what transcribe means. So outside of biology, the word transcribe means to write something down, right? You're writing something down. And this is what's happening here. We have this code written in DNA and we are transcribing it, we're writing it down. And even though I'm not going to test you on it, we'll go ahead and quickly talk about why this happens. Like, why not just have the DNA, the code from the DNA, why not just make that into protein? Well, there's a few reasons. First of all, it's in the nucleus, right? So all the translation happens outside of the nucleus. And what we don't want, that's like your one copy, right? For your cell, you have one copy of each chromosome. Well, depending on how you look at it, you have homologous chromosomes. But anyway, you've got that one copy, right? That's your master copy. So you don't want to take that DNA out of your nucleus and then take it and put it into the cytoplasm so it can be translated. That's not safe. What you don't know, and I'm not going to test you on this either, is that your cell has defenses, right? So if there's foreign DNA coming into your cells, like a virus, for example, your cells are trained to kill it and destroy it. So it's a little bit risky to have your own DNA out inside the cells because it could be destroyed. Another thing is, and I've used this analogy before, so hopefully you can remember this and it'll stick with you. You can think of your chromosomes as big books, right? Each chromosome is a big book that has instructions on how to build proteins. And remember, like this bullet point says right here, well, that's just one gene. Anyway, a typical gene is thousands of nucleotides, and for extra credit, you can look this up. How many genes are on a typical chromosome? That'd be, you look that up for independent work. That would be an interesting answer, because remember, some of them are really big, like chromosome number 1. Some of them are really tiny, like the Y chromosome. But my point is, again, you've got these books, we'll call them books, right, for this analogy. These chromosomes are books that have so many instructions on how to build all these different proteins. But if you're just trying to build one protein, you don't need the whole book, right? You just need that one set of instructions. And again, that's what transcription does. You go to the book, you go to the page that you need, and you write down the instructions that you need. You don't need the whole thing. You only need that one set of instructions. That's what transcription is. So again, I hope that helps you remember an exam. Transcription is writing something down, right? So we're going from this one language to the same language, right? DNA to RNA, same language. We're just writing it down. What you will need to know is this bullet point right here. We're going to talk a little bit more about it later. The bases of the RNA are complementary to the DNA strand. So I will give you a test question where I say something like, all right, you have a strand of DNA. This is the code. What? And then I'll say transcribe it, right? So then you'll have it's a multiple choice exam. And one of those choices is going to be correct. And it's going to have the complementary code, which, A goes with T, but there is no T in RNA, so there would be U. A goes with T, C goes with G, G goes with C, G goes with C, and T goes with A, but there is no T in RNA, there's only a U. So this would be the correct answer. You will get a test question where I give you a strand of DNA and say transcribe it. That is the correct answer. Also, and I'm going to go ahead and tell you now, so it is a trick, but I'm telling you, so it's a little bit less tricky. When I ask you that question, some of those answers, some of the choices, for that multiple choice exam, will have a T in there. So for example, one of the choices will look like this. That is not the correct answer, because remember, RNA doesn't have a T, it has a U instead. So if it's asking you to transcribe something, and it has a T in it, that's the wrong answer. Just go ahead and immediately in your brain say, that's not the right one, because RNA does not have a T. Anyway, any questions about that? Okay, moving forward, that's transcription. Translation, as you already know from outside of biology, translating something is when you're going from one language to another, right? So if someone's speaking Spanish and you can't speak Spanish, you might have a translator. Like, oh, I'll tell you what he said. You translate it. That's when you're going from one language to another. And that's exactly what this is. You're going from the nucleic acid language, which is composed of, our language, right? The English language is composed of 26 letters, right? And I don't know how many words, millions of words. The the nucleic acid language is a language that has 4 letters, right? We have 26, they have 4, right? So we're going from this language that has 4 letters, which is the DNA, RNA, nucleic acid language, to this language of amino acids. And as you know, from way back when, we've talked about it before, and we're talking about it again, there's 20 different amino acids. So this language has 20 words, if you will, or letters. Put it that way, yeah. So that language has 20 different varieties. And again, it's the sequence of the RNA bases that dictates the sequence of the amino acids of the polypeptide, which is another way of saying the letters in the RNA, those A's and C's and G's and U's, those, again, it's a language, right? Those words in that language say, bring me this amino acid. Next, bring me that amino acid. Next, bring me this other amino acid. The words in this RNA language, this nucleic acid language, say, bring me one of those 20 amino acids. Bring me that other of the 20 amino acids, right? That's how it works. And again, for the exam, I will give you a code. So maybe like, and I'm gonna bring this up later. Like that's a word, right? That is a word in RNA. CCG, that's a word. And I'll give you a chart, and I'll show you the chart later. And if you look up in the chart, CCG says, bring me a particular amino acid. So that's what's gonna happen. I'll give you a long strand of words and letters, probably about 9 of them, so three words, so to speak. And I'll say, what is the sequence of polypeptides? So again, you'll have to translate it. You don't have to memorize it. Again, I'll give you a chart where you can look it up and oh, CCG, that's this amino acid. Even I don't have it memorized, so I certainly don't expect you to memorize it. Anyway, any questions about this slide before I show you kind of a picture of what's on this slide? All right, so again, if we're looking at a DNA molecule, it's very large, there's thousands of genes on it. In this particular picture, we're focused on gene 3, whatever it may be, right? We're zooming into gene 3. We can see this code of DNA, right? That code of DNA is then transcribed, right? The bases, those letters, are transcribed into a complementary strand of RNA, right? So A goes with U, C goes with G, G goes with C. There's no T's on that one, but if there was a T, there would be an A on the other side. And again, you're gonna have a test question like that, right? That's transcription. We're still staying in the same language, the nucleic acid language. Then we go from RNA to polypeptides, which is translation. And again, this is a different language. That's why they call it translation. Each one of these 3 letters is a word that says, bring me this amino acid. So UUU in this example says, bring me a square amino acid. GGC says, bring me a circular amino acid. CGU says, bring me a triangular amino acid. Again, it's just an example. Amino acids aren't actually shaped like that, but it's just an example to simplify things. Any questions? All right, next word for attendance. You're going to want to screenshot this if you're watching the video. Tell me what shape that is. The first grade question right there. What shape is that? All right, any questions? Okay, once again, this flow of information where we go from gene to RNA to protein, it's based on a triplet code. So again, thinking of this as a language, that language of RNA is a language that only has 4 letters. Excuse me, the language of nucleic acids, whether it's RNA or DNA, right? It only has 4 letters. So for DNA, it's A, T, C, and G. For RNA, it's A, U, C, and G. So for the English language, we have everything between A and Z, right? 26 letters. This language only has 4 letters. And another thing about this language, right? Our language is interesting, right? We have short words like the word A. Those have really long words like supercalifragilisticexpialidocious. I don't even know if that's a real word, but that's a long word, right? This language, it's interesting because all of their words are only 3 letters long. And that is something you will need to know for the exam. It's based on a triplet code. So this language, each word is only 3 letters long. And again, the instructions for the amino acid sequence is written in the DNA, which is then transcribed into the RNA as a series of words called codons. You need to know that word, codons. I might ask you about codons, but I guarantee you I will use that word in questions. So codons will be used. You need to know what that means. And the best way I can describe it, other than this bullet point right here, is you can think of codons as the words. So if we're, again, looking at this like a language, again, the A's, the T's, the C's, and the G's, those are the letters. And the words are called codons. And each word is only 3 letters long. So again, it's the codon in the DNA that is transcribed into the complementary codon in the RNA. So again, as an example, GGG in DNA would be transcribed into a complementary code of CCC, right? It's complementary. It's the same language, it's just complementary. That code is then translated to make a protein, because each one of those codons, each one of those 3-letter words says, bring me this amino acid, bring me that amino acid, bring me this other amino acid. So you definitely need to know that. I'll leave it at that for now. Again, I just want to remind you, this is still an overview. We're just looking at the big picture. We haven't gotten into the details yet. If any of you are really good at math, think about this, and I'm not going to ask you yet, but think about this. If there are 4 letters in this language, and every word can only be 3 letters long, then how many words can be in that language? I'm not going to ask you that on the exam. I'll just see if anybody can figure that out. I'm not going to, don't answer it yet, but let that marinate for a little bit and see if you can figure out how many words can you make, right? You only have 3 letters, and you can only, put them in different varieties, but how many words can there be? Anyway, any questions about this slide? Again, the same picture I just showed you before, same thing, right? We have this DNA, we're zooming into this particular gene, this one part of the gene. I guarantee you that gene's not that short, but we're zooming into a part of it. You can see the code in the DNA, which is transcribed into a complementary code in the RNA. And each one of those 3 letters is a word that we call a codon, and each codon says, bring me your particular amino acid. Same stuff, right? Any questions? All right, let's do announcements and reminders. This week's lab is virtual, but you still need to attend. You should go ahead, I've said it before, I'll say it again, that is the way for the entire semester. You should assume that you need to be here. If you don't need to be here, I will let you specifically know, not as a group. You individually, I will say, hey, good, you did that, you don't need to come in. But unless you hear otherwise, specifically from me, directly to you, you should attend. Any questions about that? Yeah, good.
If I turn my lab in, do I need to separately turn my pre-lab in as well? Or can you grade them both? I'll just turn it in.
Good question. For this one, you can just turn it all in at once.
Okay.
The pre-lab, it's more like a formality. It's a great question, because normally pre-labs are very important, because I want to make sure you're prepared before you come into lab, and you're not like drawing everybody else down because you're unprepared. But because this one's virtual and everybody's doing it independently, I'm a little bit less strict on the pre-labs. So great question. But yeah, make sure you do it though. If you don't do it, you're gonna lose all those points. And it's a video. All you have to do is watch the video and answer some questions. Which again, I should remind you, if you watch a video and answer questions, and it asks for your name and your course number, please use your full name and the correct course number, which is 101-02, or even if you want 101-2. Either one of those, because when I go to grade them, it's a big spreadsheet, so I hit a search function. If I'm searching for 101-02 or 101-2, and you put 101-3, then I'm not going to see that you did it. You're going to get 0 credit. Anyway, just as a reminder, I've been saying this for about a week now, but I'll remind you again, for those of you who are here in person, you're getting now 4 points of extra credit instead of 2. So again, I'm trying to bribe people to attend. And of course, if you're online or watching the video, it's still just 1 point for extra credit. Any questions about that? Okay, exam 2, still open for a retake. This blows my mind. Nobody is asked to retake it. No one retook exam 1. But I guarantee you, someone at the end of the semester is going to be like, is there anything I can do about my grade? I can't get a D, I can't get an F. I'll be like, why didn't you retake the exams? All that. So I'm telling you now, do this stuff, please. There's nothing to lose. You can retake the exam as many times as you want. If you get a lower grade, just don't tell me about it. I won't adjust your grade. When you get a higher grade, tell me about it, and I'll go adjust your grade. It's open book, right? There's nothing to lose. Likewise, independent work. Remember, that's 100 points. It's a full letter grade. It's due in about 6 weeks. You should start working on it. If you haven't done a paper yet, you should at least do one paper so you can see how I grade. That's a full letter grade. Again, someone at the end of the semester is like, is there anything you can do about my grade? And my answer to me, no. I've been telling you to do something about your grade this whole semester. That's completely in your control. It's not exams, it's not labs. Just write something. Write about whatever you want. Just keep turning things in until you hit 100 points. Or even more. One of your classmates has more than 100 points. And anything past 100 is added to her extra credit total. Anyway, any questions about independent work? All right, it is now too late to drop the class. That was Friday, was the last day to drop the class. So if you're still here, some of you really need to turn things around. I remind you, I'm here to do it. Like some of you should almost like start fresh in a sense. Like meet with me, let's figure out the days that you've missed. And you can go back and watch the videos to get caught up on the lectures. Let's talk about the labs that you've missed, the lab reports that you've missed, all that. I can help you find a path, so to speak. That's the first part, right? Is finding out what you need to do. I'll help you find out what you need to do, and I'll help you do it. I'm here to help. And then finally, like I've been saying for a while, lecture and office hours the Monday after Easter and the Friday before Easter will be 100% online because I will not be here. Any questions about announcements and reminders? Do you guys have any announcements or reminders? Okay. The next word for attendance then is to tell me, is it too late to drop this class? Can you still drop this class, in other words? So, I guess the next phrase for attendance will be multiple choice. You either say, it is too late to drop this class, or it is not too late to drop this class. That's your next phrase for attendance. All right, any questions? All right, let's get some more background information. We're still doing an overview. We haven't even gotten into specifically how transcription works and how translation works, right? I'm giving you all this. First, I just gave you that overview. Now I want to get a little bit more into the nitty-gritty about the genetic code, right? So you already know, I've already told you some of it. I'm going to repeat myself here again. But you know about like these words are 3 letters long, and that's called a codon, and it's complementary, all those things, right? So we're going to, again, dig deep into that before we get into this. So some of this is going to be a review. So again, the genetic code is a set of rules, if you will, a set of words. It converts RNA sequence into amino acid sequence. So here's that question I was asking you earlier. There's the answer. In this language, there's only 4 letters, right? And the words are 3 letters long. So that being said, how many words can you make? And the answer is 64. So what you don't need to know for the exam, which is why I have a star next to that, but I'm going to go ahead and tell you, you don't need to know this, but just so you know, of those 64 words in this language, 61 of them are for a particular amino acid, right? Like, so bring me this amino acid, bring me that amino acid. 3 of them are called stop codons. And that says, all right, we're done with this. That's the word that says stop the process. That's what a stop codon is. You need to know what a stop codon is. You don't need to know that there's 3 of them. But the point is that some of these words in this language say stop. Hopefully, if you guys are paying attention and really thinking about things, I'm hoping what's on the screen right now is a little bit confusing to you. Because, like I said, there's 64 words, or let's just say 61. There's 61 words that say, bring me this amino acid, bring me that amino acid. But how many amino acids are there? And you guys do need to know this. Does anybody remember yet? How many? I think you said 20? Yeah. There's 20 amino acids, right? So hopefully that's you're probably like, what the hell? 61 words that say, bring me this amino acid, bring me that amino acid, but there's only 20 amino acids. Hopefully that'll tell you that some of these words are saying, bring me the same amino acid, right? That's what this next slide is. This is a chart I will give you on the exam, and I will give you a code and ask you to translate it. So if I say CCC, right, and I say translate that, you would start here and be, okay, C, C, C. And that says bring me the amino acid proline, right? And then if I said, I don't know, GCA, okay, G, C, A, alanine. So I would give you, this will be on the exam, and I will ask you to translate a string of letters, and you're gonna tell me which amino acids, which amino acids is supposed to be there. So again, nothing to memorize, I'll give this to you. And there will be at least one question where I give you a strand of DNA, right? So you have to go from DNA, I'll give you those letters in the DNA, and you have to figure out what it is in RNA. Once you figured out what the RNA letters are, then you can use this to figure out which amino acids it's coded for. Anyway, any questions about that side? All right, like I mentioned before, not that I'm gonna test you on this, but to me this is so interesting. The genetic code is universal. It's shared by the simplest bacteria to complex plants and animals. So I put a star next to universal because there are some exceptions. Not that you need to know that. You can look it up for independent work. What are the exceptions? But yeah, for the most part, it's universal. So we can take a human gene, stick it in a bacteria, and that bacteria will produce whatever it is that gene calls for, whatever protein that gene calls for. And you can look that up for independent work too. There's some really interesting things out there. I think when I was a student, I remember one of the newer stories was they took a gene for the spider's silk, right? The protein to make the spider silk. So they took that gene, I think they put it in a cow or a goat or something. And then when that animal produced milk, the protein for that silk protein, for that silk, the spider silk, was in that goat's milk, which is interesting to me. And of course, it's not just interesting random things like that. Some of them actually have a purpose. For example, if you know anybody who's diabetic and they take insulin, this is where that comes from. It used to come from pig spleens, right? They used to kill a bunch of pigs and take the insulin from their spleens. Now, we take that human gene that says this is how you produce insulin, and we put it into bacteria because bacteria are really small and they're easy to grow. And those bacteria make this human insulin. And then that's where we get it from. Anyway, very, very interesting. It's an interesting thing to look into for independent work if you want. It's also interesting to think about just the world of possibilities that have not been done. Like for example, I want to one day move to Washington and figure out how to get the gene that produces THC, put it into foods that we eat, right? So, instead of smoking pot or eating edibles, you're like, hey, I had salsa, but this tomato had THC in it. I think that'd be cool. So again, that's the kind of stuff you can look into for independent work. Anyway, here's some examples now that I'm going to ask you. Can anybody see what's different about that middle pig? It's not a problem with the picture. It's not an issue with the projector. That pig actually does look different. Can anybody see what it is? That's okay. It is a little bit hard to see. It's glowing. Its nose and its hooves are glowing because somebody took a gene that they got from a jellyfish, the one that makes them glow, and they put it into that pig, and now that pig glows. Which sounds random, but there actually are purposes for this. Same thing with that on the bottom right. That is a mouse or a rat or something. It's a baby something, a baby rodent, and it is also glowing. Its arms are glowing because again, someone put a protein from a jellyfish into that animal. This is not a real picture, but I've seen it shared online, so I thought I'd share it. It's really possible. As far as I know, no one's done it. What they're suggesting there is take that same gene that makes jellyfish glow, or in this case, pigs and rodents, put it into trees. That way, trees glow, and you don't have to have street lights. So yeah, it's possible. No one's done it, as far as I know, but it is possible. Of course, there would be bad implications, because animals need trees to survive, and if they were glowing, that would probably throw off the ecosystem because those animals make, what the hell is a glowing tree? I'm staying away from it. But hey, better than a streetlight, I guess. Anyway, we'll move forward. That's it. That's all the background information you need to know. Finally, we can get into the nitty gritty and start getting into the details. And let's talk about transcription. Again, transcription is the transfer of genetic information from DNA to RNA. Or if you were making a flashcard to study, on one side you could just put transcription, on the other side you could just put DNA to RNA, right? You need to know the transcription is going from DNA to RNA. What I haven't taught you yet, this first bullet point is new. The DNA opens itself up. Well, it doesn't open itself up, but it gets opened. Something opens up the DNA so it can read the code. Which sounds familiar, right? This is a lot like DNA replication. Remember, I was saying to make this new strand of DNA, this DNA polymerase attaches to the DNA and it opens it up so it can read the code, and then it brings in all these new nucleotides. The same thing is happening here. We have this enzyme that opens up the DNA, it's reading the code, and it's bringing in all these new nucleotides, except in this case, instead of being DNA nucleotides, it's bringing in RNA nucleotides, which again are the same, other than the fact that RNA does not have a T, it has a U instead. So yeah, in a sense, as far as we're concerned in this 100 level non-biology major class, transcription is a lot like DNA synthesis, DNA duplication, DNA replication, however you want to say it. Because, like I said, it's very similar. We're opening up the DNA, we're reading the code, we're making a new strand of nucleic acids by bringing in one nucleotide at a time. So here's a question for you if you want to look this up for independent work. What happens? Like if, let's say the machine is doing this, it's going from DNA to RNA, the machine, the cellular machinery is doing transcription. When it's bringing in all these different nucleotides one at a time, what happens when that machine tries to bring in a T? Because remember, RNA doesn't have a T, right? RNA has a U instead. So what happens? Like, does that cellular machinery, does it just know not to bring in a T? Or is there some kind of way where if it brings in a T, it recognizes that it shouldn't be there, so it gets rid of it? It's things you can look up for independent work. Here's a question for you that I am actually asking. This is not an independent work question. Does anybody remember the name? Forget about this for a second. Does anybody remember the name of the enzyme, the cellular machine that makes DNA? Remember this chapter, we started talking about how DNA is duplicated during S phase of interphase. And I told you the name of the enzyme, because remember, it's named after what it does. Does anybody remember the name of that enzyme? Very good. It has a polymerase in it, right? Because that's its job. It's making a polymer, right? So polymerase is the second part of the name. Do you remember the first part? DNA. Good. Perfect. DNA polymerase. You need to know that for the exam. DNA polymerase is the name of the enzyme that makes DNA. With that being said, that's on the next slide. I'm going to show you this. Let's see if anybody can guess it. What do you think the name is of the enzyme that does this? What do you think the name of the enzyme is that makes the RNA? And it's not a trick question. This is one of those few times where biologists they chose nice words that actually made sense. So DNA polymerase makes DNA. RNA. There you go. RNA polymerase. You need to know that for the exam. Yes. RNA polymerase is the cellular machine that does it. That is the enzyme. And again, it's an enzyme, so it ends with ACE. And enzymes are named after what they meant, their substrate or what they do. In this case, it's named after what it does. It makes an RNA polymer. And once again, RNA does not have a T, it has a U instead. So again, any question where I ask you to transcribe something, which is just a different way of saying, hey, make a strand of RNA. If I ask you to transcribe something with those in this multiple choice exam, anything with a T in it is not the correct answer. Because RNA does not have a T, it only has a U. Anyway, that's what it looks like. Any questions about this slide? Okay, another thing I want to point out is this is a 100 level class for non-biology majors, so this is oversimplifying things. You want to look into it for independent work, look into RNA polymerase. It's not that simple. That's why I like this picture. Notice that it has multiple colors. It's because actually RNA polymerase is a team of proteins. It's not just one protein. It's a bunch of proteins working together. So that again, this is the simplified version. For independent work, you can look into the more detailed version. What are all the parts that actually do it that you guys don't need to know about? Anyway, here's another simplified picture of our RNA polymerase. Very simplified, that it works for this class. Again, you can see here the cellular machinery, if you will. The RNA polymerase is attached to the DNA. It's opened it up so that the code can be read, and it brings in one nucleotide at a time, right? The complementary nucleotide. There's a T, so it brings in an A. There's an A, so it brings in a U. There's an A right here, so another U is coming in. It brings them in, and makes these covalent bonds between them, and it creates this new strand of RNA. Any questions about that? All right, now let's make things more difficult. Because Lord knows we can't have things be too easy. So like I said, the first thing you need to memorize is the fact that we go from DNA to RNA to protein. After you memorize that, then memorize the fact that the process of going from DNA to RNA is called transcription. Once you have that memorized, memorize the fact that transcription is done by RNA polymerase. Once you have that memorized, I'll throw this in there for you. Now you need to know that transcription itself is broken down into 3 parts. Initiation of transcription, RNA elongation, and then termination of transcription. You need to know that. You need to know it in that order. You're going to need to know what happens at each one of those stages, and I'm about to teach you that. And you can see here I bolded initiation, elongation, termination. Because if you have that memorized, then you're fine. I'm not going to get tricky with it and like have different versions of initiation or different versions of elongation or different versions of termination. If you just remember initiation, elongation, and termination, you're good. The good news is, in my opinion, this is actually pretty simple because it's in the name. Also, it's in in this order. It has to be this way. Like, what do you think initiation is? I mean, it's probably the question's so easy. It probably sounds like a trick question. So I'll just say it. Initiation is when the process starts, right? Hence initiation, when you initiate something. That's when it starts. RNA elongation is obviously the RNA getting longer and longer. And the termination is obviously just when it ends. The whole process is terminated.
Wait, when does it stop? When does it stop on the script?
Termination is when it stops.
Okay, well, yeah, in what, like,
Oh, good. We'll get to that. So hold on to that question. Very good question. And that question will be answered. But yes, first memorize. Those are the steps. That's what they're named. So we'll talk about them just separately. So when we get to the end, we'll talk about what you're asking. So one of the things you need to know about transcription is that it starts at a place called the promoter. So it's kind of like the opposite of what you're asking. Instead of asking, you're asking where it stops. I'm telling you where it starts for now. So you need to know this. It is called the promoter. And I'll go ahead and skip ahead and tell you. Later, when we talk about translation, we're also going to break it down into the 3 steps. And you're also going to need to know where it starts. So for this, it's different, right? So for transcription, it starts at the promoter. Translation, it starts somewhere else. You'll learn that when you get there, but you will need to know the difference between the 2. So yes, transcription starts at a place called the promoter. This is located at the beginning of a gene, and it is where the RNA polymerase attaches. So the cellular machinery, that enzyme, it reads that code and says, oh, that's me. That's where I need to attach. So initiation, like I already said, this is where it starts. But if we're going to get a little bit more specific, initiation is technically when the RNA polymerase attaches to the promoter. That's it. That's initiation. The RNA polymerase, the enzyme that's going to make the RNA, attaches to the DNA at a place called the promoter. This picture is not from your textbook, which is why you see things that you might not recognize, like 5' and 3'. We don't worry about that. Actually, I think your textbook might even talk about 5' and 3', but you don't need to worry about that. Yeah. Anyway, any questions about promoter or transcription in general? Because this is it. This is the whole conversation about, initiation. Next, we're going to talk about the second stage. All right, the second stage is elongation. Again, it's in the name, right? Elongation of RNA. The RNA is getting longer. That's it. So again, the RNA polymerase is reading the DNA code and is bringing in these covalent bonds. Excuse me, it's bringing in these nucleotides and covalently bonding them to each other. It's another independent work question for you. Obviously, we're building proteins here. Or it's good grief, I can't talk today. We're building molecules, right? Every time we bring in a new nucleotide and covalently bonded to the strand, that's building a protein. Or good grief, building a molecule. So the question I'm asking you, if you want to look into it for independent work, is where do we get the energy for that? Obviously ATP, right? But how much energy is used? How many ATP are used for every one nucleotide, or every two nucleotide, or every three nucleotides? You can look that up if you want for independent work. Anyway, so the only other new thing here on this slide, like I said previously, the old part that I've already told you is that this machinery opens up the DNA so the code can be read, and then once it's done reading it, it closes it back up. And again, there's other RNA polymerases. This is oversimplified. There's other proteins involved, but we're just not getting into it. So you could look that up, any of this up for independent work, and get the true story instead of just this simplified version of the story. So like, yeah, what enzyme does open up the DNA? What is that thing called? If you want to look it up. Anyway, that's it for elongation. Any questions about elongation before we talk about the third part, which is termination? And finally, we'll be able to answer Lebby's question. All right, third part is called termination. To answer Lebby's question, where it happens is a place called the terminator. So that's nice and easy, right? Termination is when the process stops, and it is at a place called the terminator. So the terminator, in a sense, is the opposite of the promoter, right? The promoter is where the RNA polymerase attaches to the DNA. It says, all right, this is where I attach. And then it moves down the DNA and it reads the code and it reads the code and it reads the code. And then finally, when it gets to the terminator, it says, whoa, that's me, I'm done. This is my signal to stop. And then it detaches. And of course, as we already mentioned previously, during elongation, at the end of the elongation, the DNA does come back together. So obviously here the same thing happens. The DNA comes back together. So any questions about termination? Excuse me. All right, that's nice and easy. And again, I'll remind you, later, probably Wednesday, we're going to talk about translation. And translation also has 3 stages. It also happens to be called initiation, elongation, and termination. But the words are going to be different. So the start place and the stop place and the cellular machinery, all different names. So keep that in mind when you're studying this. So what I'm teaching you now is all about our transcription. So you need to know that. Not only do you need to know it, but again, later you're also going to need to be able to differentiate between transcription and translation. Anyway, if there's no questions, I'll show you a picture of everything we've talked about, the whole thing. So here we have this long strand of DNA. It's a gene. The RNA polymerase recognizes the code in the promoter DNA. That's where it attaches, right? It opens up the DNA, as you can see here, so it can read the code, and it's building a strand of RNA, and it moves down the DNA, reading the code, making the strand longer and longer, until finally it reaches the terminator where it says, oh, that's my code, I'm done, and it releases. The RNA polymerase releases from the DNA. The RNA itself releases from the RNA polymerase. So all the parts go their separate ways. Another thing you can look into for independent work, and again, keep in mind I'm oversimplifying this, but RNA polymerase, whether we're looking at it as one thing or the truth, which is it's a bunch of things, is it reusable? So this RNA in this picture, this RNA polymerase, now that it's done with its job and it's detached, does it just go float around and do nothing? Or does it do its job again? Is it reusable? Something you can look into for independent work. Let's see. The next word for attendance is going to be a color. There's 3 colors on here. The DNA strand, anyway. The DNA strand is mostly blue, but it also has a little bit of green on it and a little bit of red on it. So the question is, what color is the terminator? Is it blue, green, or red? That's your next word for attendance. And again, if you guys are here in person, you don't have to send those words, but you can to get extra credit. If you're online, you have to send those words before 9 a.m. And if you're watching the video, you have to send the words in addition to a screenshot when I said the words. Anyway, any questions about this slide? All right, moving forward. If you download the PowerPoint, you can watch this little animation that shows it happening, transcription. 5 minutes? No, this is perfect. We can talk about this. So again, transcription is going from DNA to RNA, right? We've talked about that. You know how it happens. But now let's talk about actually processing that RNA. That's not the end of the story. That RNA is not ready to leave the nucleus and go get translated into proteins yet. Now in prokaryotes, they don't have a nucleus. So for them, this is different, right? So for them, when they transcribe this DNA into RNA, it goes directly, it's translated directly, like right away, that RNA is then translated into a protein. But for everything else, for us eukaryotes, right, so plants, animals, protists, things like that, fungi, we're more complicated, right? We do have a nucleus. So that RNA is not ready to go yet. And here's a new thing I'm teaching you, and you're going to need to know this. This particular type of RNA we're talking about is called mRNA. This is the messenger RNA. You need to know that. Later in this chapter, I'm going to be giving you different types of RNA, and you're going to need to know the difference between them. So messenger RNA, it's in the name, right? It's got the message. Notice too, by the way, that messenger RNA is made from DNA. And, like I've already said, and now I'm just giving you the word for it, mRNA. mRNA is translated into proteins. So when people say that the vaccine, the mRNA vaccine is gene therapy, you just know they're dumb, because gene therapy is editing your genes, editing your DNA. mRNA doesn't do that. mRNA is just a code that says this is how you build a protein. So again, like I said, remember, we go from DNA to RNA to protein. We don't go from RNA to DNA. There are some exceptions. You'll learn that later. But yeah, anyway, that's what mRNA is. You need to know that for the exam. So again, in eukaryotes, this mRNA needs to go through some changes. And that's what we're about to talk about. The first thing you need to know about is this thing called cap and tail. So that RNA, we have the RNA, and then we add some stuff to it. We add a cap to it at the 5' end. Not that you need to know the 5' part. We add a tail to it, which is just a bunch of A's at the 3' end. Not that you need to know the 3' part either. What you need to know is what cap and tail is. Then again, it's we're adding stuff. We're adding more nucleotides to both ends of it. So you might be wondering why, and I probably won't ask you this, but the reason why, like I said before, when your cell, like when you have foreign DNA or foreign RNA in your cell, it knows to destroy it, right? Because that's how you get viruses and infections. So your cells attack foreign DNA, they attack foreign RNA that shouldn't be there. So this just protects it. This helps it say, okay, your cell's like, all right, this is supposed to be here, let's not destroy it. It also helps it attach to the machine that does the translating, which I haven't talked to you about yet. It's called a ribosome. You're going to learn about it later. But the ribosome can recognize it by that. So another thing for independent work, if you want to look into it, is how does this even happen? What cellular machinery adds the poly-A tail? What cellular machinery adds the cap, right? So you know RNA polymerase makes the RNA, but what does all this? You don't need to know it for the exam, so you can look it up for independent work. What does the cap and tail? And yeah, I guess we can end it there. When we come back, we'll finish talking about its processing of eukaryotic RNA. And the next thing we'll talk about is RNA splicing. But for now, that's it. So the last word for attendance. Let's leave it at that. So yeah, don't forget, the last phrase for attendance is to ask me a question about the material presented in this lecture. You have to it has to be about this for grading purposes. Now, you can ask me whatever you want, just for the sake of questioning, but for the sake of grading, you need to ask a question about the material. That's it. See you guys on Wednesday. Write some papers, retake some exams. And don't forget the final exam, half of it's based on the first 2 exams. So it would really help you, even if you don't even care about your grade for exam 1 and exam 2. Go in there and retake those exams just to get the practice, because that'll help you for the final.
Have a good day.
You too.